Look closely at this DNA unzipping. You might expect two new strands, but instead, just a single RNA strand emerges from the bubble. This is transcription, the selective process of copying localized genetic information from a piece of DNA into a mobile RNA molecule. It is easy to confuse this with DNA replication, but they operate under completely different biological rules. When a cell replicates, it duplicates the entire genome. Transcription only ever copies a precisely defined segment. The machinery also strictly copies only one of the two DNA strands. If both strands acted as a template, they would code for different RNA sequences. A single segment of DNA would end up coding for two different proteins, tangling the cell's information transfer system. Furthermore, those two simultaneous RNA molecules would be complementary to each other. They would immediately lock together to form a useless brick of double-stranded RNA, making it impossible for ribosomes to translate them into protein. To understand gene expression, we must discard the idea of total replication and examine the exact biological hardware that enforces this asymmetrical copying. The first logistical problem a cell faces is navigation. DNA is a massive molecule, and the machinery needs physical boundaries to know exactly where a specific gene starts and ends. This diagram shows a transcription unit. It is defined primarily by three distinct regions: the promoter block at the start, the structural gene in the middle, and the terminator block at the end. Notice the physical orientation of these two DNA strands. They are anti-parallel, meaning they run in opposite polarities, one from 5' to 3' and the other from 3' to 5'. This geometry dictates the entire process because the enzyme doing the work, RNA polymerase, has a strict mechanical limitation. It can only catalyze polymerization in one continuous direction, 5' to 3'. To build a new RNA molecule in that 5' to 3' direction, the polymerase must read the opposite DNA strand. That means the strand with the 3' to 5' polarity becomes the template strand. Meanwhile, the opposite strand, running 5' to 5', is pushed aside and temporarily displaced during transcription. Biologists call this displaced strand the coding strand. It sounds paradoxical since it is not read by the enzyme, but its sequence is identical to the newly minted RNA, simply swapping out thymine for uracil. The physical anchor that dictates all of this is the promoter. Its location at the 5' upstream end of the structural gene determines which strand the polymerase binds to, and therefore which strand becomes the template. If you were to physically switch the positions of the promoter and the terminator, the system would run in reverse. The definition of the coding and template strands would flip. The strict geometry and polarity of the transcription unit serve as biological safeguards, guaranteeing that genetic instructions are always read in the correct direction. In bacterial cells, this reading process operates without physical borders. There is no nucleus compartmentalizing the cell interior. Bacterial systems are built for efficiency. A single type of DNA-dependent RNA polymerase is responsible for synthesizing all three major types of RNA: messenger, transfer, and ribosomal. However, this core bacterial polymerase has a specific mechanical constraint. Left on its own, it cannot initiate or terminate the reading process. This animated diagram illustrates how bacteria solve the problem. The polymerase requires a transient modular plugin called the sigma factor to jumpstart transcription at the promoter site. Once the enzyme successfully opens the helix and begins elongating the RNA strand, the sigma factor drops off. When the polymerase reaches the terminator region, it relies on a secondary plugin, the Rho termination factor, to detach the nascent RNA and halt the sequence. By associating transiently with these separate factors, the cell alters the specificity of the polymerase exactly when needed, rather than building a single, overly massive enzyme. Because bacterial messenger RNA requires no processing, and because there's no nuclear wall in the way, ribosomes often attach to the mRNA and begin building proteins before transcription is even finished. The bacterial transcription model prioritizes rapid, continuous execution over data security. This coupled mechanism is possible only because the cell lacks internal physical barriers. Human cells operate under a much stricter architecture. Plunging through the membrane into the crowded, enclosed environment of the nucleus, we find the genetic data physically walled off from the rest of the cell in tightly packed chromatin. This environment demands a clear division of labor. Instead of a single enzyme, eukaryotes rely on at least 3 distinct RNA polymerases. RNA Polymerase II is the specific engine tasked with transcribing the precursor to messenger RNA. The direct output of this enzyme is called heterogeneous nuclear RNA, or hnRNA. This is a raw, unedited initial transcript. This diagram shows exactly why hnRNA requires editing. Eukaryotic genes are split. The sequence of information on the DNA is deeply fragmented. The actionable, expressed sequences that actually hold the code for building proteins are called exons. These exons are heavily interrupted by introns, intervening sequences of genetic junk data that serve no purpose in the final instructions. If a ribosome were to immediately translate this raw strand, the presence of those introns would cause a catastrophic misread, producing a non-functional protein. The shift to a compartmentalized nucleus forced eukaryotic cells to separate raw data generation from data execution. To bridge that gap, the cell executes an extensive editing process. Before any editing can occur, the raw hnRNA must be chemically armored to survive the journey out of the protective nucleus. First comes capping. The cell attaches an unusual nucleotide, methylguanosine triphosphate, directly to the 5' end of the transcript. Next is tailing. The machinery chains 200 to 300 adenylate residues to the 3' end in a template-independent manner. This cap and tail armor prevents cellular degradation mechanisms from destroying the transcript while it is in transit. With the molecule protected, the cell deploys its splicing mechanism to act as the final molecular editor. Splicing targets and systematically removes all the non-functional intron segments from the sequence. Finally, the remaining functional exons are fused together in a strictly defined order to create a continuous, readable code. Only after capping, tailing, and splicing are complete is the molecule officially classified as mature messenger RNA. It is a refined set of instructions, ready for translation. The mature messenger RNA molecule now slides smoothly out of a nuclear pore, leaving the nucleus and entering the expansive fluid of the cytosol. Despite vast differences in complexity, both bacterial and eukaryotic transcription rely entirely on the strict asymmetric geometry set by the promoter. The difference lies in an evolutionary trade-off. Prokaryotic systems prioritize the raw speed of a single compartment, while eukaryotic systems prioritize careful, compartmentalized data curation. The bizarre presence of introns and the absolute necessity of splicing are architectural relics. They point back to an ancient RNA-dominated world, one that is still operating as the core engine inside every human cell.